In this video, let's discuss about animal kingdom. There are millions of animals on earth. So classifying them and arranging them in a systematic portion will help in the easy identification of animals. So what are the basis of classification? Actually, classification is based on grouping the animals based on similar characters like cellular organization, symmetry of the body, pattern of digestion, circulatory and reproductive system. And this classification will quicken the work as well as it will avoid the confusion. First of all, let's see what is levels of organization. So living organisms are made up of cells. So millions on trillions of cells combine to form an organism. The tissues will join to form the cells will join to form tissues. Various tissues will group together to form organs, and when various organs are grouped together, it will turn into an organ system like digestive system, circulatory system, reproductive, respiratory systems, like that. So the cells will group to form tissues, tissues will group to form organs, organs will group to form organ system. This is known as levels of organization. So in a digestive system, there are two types. One is incomplete, another is complete. So in the case of incomplete digestive system, which is found in primitive organisms, that is first formed organisms, there will be only one opening in the body that serves both as mouth and the anus. For example, in platyhelminthes. In complete digestive system, there will be two separate openings, one for mouth and another for anus. The circulatory system is also of two types. One is open type, another is closed type. In the case of open type, there is the blood will not be circulated through heart. It is pumped out of the heart, and the cells and tissues are directly bathed in the blood. The heart or heart-like organ will pump the blood out of it. There is no tube-like structures that is carrying the blood, whereas in closed type, vessels will be carrying blood in and out of the organs. The vessels are grouped as arteries, which will carry pure blood, veins, impure blood rich in carbon dioxide, and capillaries. In the organs, the arteries and veins will be branched as capillaries for fine distribution of blood. The next comes symmetry. Symmetry, if we take an organism and if we draw a plane or a line passing through it, that is known as symmetry. So if the symmetrical line is if a symmetrical line is drawn in any angle, if we are not obtaining any equal position, equal halves. For example, if I cut from this side, these two sides are not equal. If I cut from this side, these two are unequal. So wherever I draw the line, I am getting an unequal half. This kind of symmetry is known as asymmetrical. Example, sponges. So wherever I draw the line, I will never obtain a equal half. This is known as asymmetrical organisms. Example, sponges. In the case of radial symmetry, wherever I draw the line, I will get two equal halves. So through an organism, here it's given, through an organism, if I draw a line through any angle, I will get equal half of the body. That is known as radial symmetry. In the case of bilateral symmetry, if I draw a line, for example, let's take human. So two hands here, two legs here. So if I draw a line through this direction, I will get two equal half of the body. But if I draw like this, I will never obtain a equal half. So only only in one plane I am getting two equal halves. So this kind of symmetry in which we will get an equal half of the body in only one plane. Example, annelids arthropods. So example given in the book is crab. If a line is drawn like this, we will be getting two equal halves. Whereas if we change in any other angle, two equal halves will not be obtained. So this is the case of symmetry. Asymmetry, the plane will never cut the body into two equal halves. Radial symmetry, in any angle the plane will cut the body into two equal halves. Bilateral symmetry, in only one direction the plane will cut the body into two equal halves. Other directions it will be unequal halves. Next is diploplastic and triploplastic organization. So these are some of the characteristics used for classification. So what is diploplastic organization? So during the embryonic development, two layers will be formed. One is ectoderm outer, inside is endoderm. If the embryonic layer is composed of two, it is known as diploplastic organization. In between this ectoderm and endoderm, an undifferentiated mass of cells called mesoglia will be present. So this is known as diploplastic. Only an ectoderm and endoderm will be present. Example, coelenterates. Triploblastic, three layers will be present in the embryonic development. One is ectoderm, center mesoderm, and inner endoderm. Example, platyhelminthes to chordates. So if only ectoderm, endoderm is present, it is diploblastic. Ectoderm, mesoderm, endoderm is present, it is triploblastic. In the case of diploblastic, ectoderm and endoderm are separated by an undifferentiated layer known as mesoglia. Next is coelom. Coelom is actually an empty cavity. Cavity is an empty area. If the cavity is lined by mesoderm, it is known as coelom. If the organism has coelom, it is known as coelomates. Example, annelids, mollusca, arthropods, echinoderms, hemichordates and chordates. So let's see three in this. So here we have a cavity, ectoderm, mesoderm, endoderm. Here the cavity is lined by mesoderm. So this is coelomates. And in this case, ectoderm is there, endoderm is there. Here the mesoderm is present as patches. It is found as patches here and there in the cavity. This kind of coelom is known, organism possessing this kind of coelom is known as pseudocoelomates. Actually, pseudo means false. So if the mesoderm is found as patches in the coelom, it is known as pseudocoelomates. Example, Ashelminthes. If the coelom is completely absent, ectoderm, mesoderm, endoderm is there, but the coelom is absent, this kind is called acoelomates. Example, platyhelminths. So if the organism is having a cavity which is lined by mesoderm, it is known as coelomata. If the organism is having mesoderm as patches in the cavity, it is pseudocoelomates. If the coelom is completely absent, the organism is known as acoelomates. Next is segmentation. So the body will be divided into various segments inside and outside. For example, earthworm. The segmentation found in the body of the earthworm is known as metameric segmentation, and the scientific term for this is metamerism. And notochord. During embryonic development, there will be on the dorsal side of the embryo a prominent or stiff structure will be there, which will later develop into the brain and the spine, and this is known as notochord. It is a rod-like structure found on the dorsal side of the embryo. Later it will develop into brain and spine. Example, chordates, and it is present in the embryo. So if this notochord is absent, those organisms are known as non-chordates. Example, porphyra to echinodermata. Actually, these are advanced. If the notochord is well developed, that is an advanced one, and these are primitive one. It's very easy. If the organism is having a spinal cord or vertebral column, that is chordates. If the vertebral column or the spinal cord is absent, those organisms are known as non-chordates. So here everything is summarized. So far what we have seen, it is summarized like this. So the main kingdom is Animalia. So cellular level, if it is made up of single cells or a group of cells performing some function, that is cellular. And if the cells are grouped together into various tissues and organ system, it is divided into cellular level and tissue organ level of organ system level. So cellular level, very primitive. Example, porphyra. Here based on symmetry, radial symmetry, bilateral symmetry, in the cellular level only we can see that asymmetry. Example, amoeba, if you cut a plane in any angle, you can never obtain an equal half, so it is mostly asymmetrical. In here based on symmetry, it is of two types. One is radial, another is bilateral. Only in one plane, in any plane you will get a two half. Here in only one plane you will get a two half. And if the radial symmetry means coelom will not be present. Example is Coelenterata and Ctenophora. So about this, we will discuss in detail in the upcoming videos. In the case of bilateral symmetry, if the body cavity is absent, coelomates, pseudocoelomates is also there, coelomates is also there. Radial means only acoelomate will be there, that is coelom will be absent. But in the case of bilateral symmetry, all the three categories of the coelom will be there. Accordingly, acoelomates example is platyhelminthes, pseudocoelomates is aschelminthes, coelomates Annelida, Arthropoda, mollusca, Echinodermata, Hemichordata, Chordata. This means coelomate is the advanced one compared to these three things.